Okay, so now we are moving on to looking at purine and pyrimidine metabolism. These, of course, are important in RNA, DNA, important for energy sources, and so their formation inside of the body is very important, but also they're different. So purines are going to be formed, but also have a lot of recycling, and it's because it takes a lot of energy to form those. Pyrimidines are also going to be formed, but it doesn't have as much control and regulation as your purine synthesis. So we'll talk a lot about the synthesis of these first, and then we're going to talk about how they're degraded. Purine degradation can lead to some diseases, whereas pyrimidines are more limited in the diseases. They typically get degraded into products that can be easily either reused or excreted. And then we'll talk about salvaging purines. So again, that's about recycling purines, and that's an important process. And if you have defects in those salvaging pathways, they can lead to severe disorders. And so keep in mind that purines and pyrimidines are going to be, of course, bases in DNA. So cells that rapidly divide are going to need more of these than cells that are more just metabolically active but not dividing. Okay, so just as a big overview, again, these are going to be required for making our nucleotides as well as our nucleic acids. They can be synthesized from scratch. So you have a few ways to make these. So synthesis from scratch, or they can be salvaged. And this would be from existing bases. Now, when you're making your purines, these are going to form, you're going to form two rings. So your purines have two rings, and your pyrimidines only have one. Now your rings, I should actually, let me move this to make it just a little bit easier. Okay, so your purines have, two rings. Your pyrimidines have one ring. Now to make the purines, it is actually an imidazole ring. Plus a pyrimidine ring. Okay, so that's going to form your purines, whereas a pyrimidine ring just has a pyrimidine ring. You can also take these up in the diet, but most of the nucleic acids that are taken up in the diet are going to be metabolized by those cells taking them up, the intestinal epithelial cells. So you have your so you actually don't absorb very much of what you actually take in from your diet. Now, the de novo, or synthesis from scratch of purine synthesis, is going to be very complex. So it's a complex process, highly regulated. It's going to have 11 steps. It's going to require 6 ATPs for every purine that's made. And the precursors are shown here. So we have our precursors, we have our glycine, we have our ribose 5-phosphate, glutamine, we have some carbons donated from our tetrahydrofolate pool. We have nitrogens that are going to come from aspartate. Glutamine is going to donate some nitrogens. And we have two different places where our tetrahydrofolates will add. And then also we have a carbon that's going to come from carbon dioxide. Now, purines are going to be synthesized with the initial purine being inosine monophosphate. So the first purine, so for synthesis, or IMP. Adenosine monophosphate and guanosine monophosphate are each going to come from IMP, and they have two different reaction pathways to do this. So from inosine, you'll get AMP and you'll get GMP. Now, the enzymes in this pathway are going to be deaminases, kinases, phosphorylases, phosphorylase and you also have a hypoxanthine guanine phosphorylase involved in this. So there's a lot of things involved in your purine synthesis. The Purine Nucleotide Salvage pathways is going to allow you to take free purine bases and convert them into nucleotides, and then go from nucleotides into nucleosides, and then back to free bases. So this is just an example of a salvage. So in your salvage, you can go from your nucleotides. Let's see. So we have our purines to nucleotides, and then from our nucleotides into nucleosides, and then from our nucleosides into our free bases. And there's going to be interconversions between your adenosine, guanine, and your inosine with respect to the salvage pathways. Now, I just want to mention here the difference between a nucleotide and nucleosides. So a nucleoside is shown here. A nucleoside is basically your sugar, it can be ribose or deoxyribose, with a base. So sugar plus base is a nucleoside. And as you start to add phosphate, this is when you get to your nucleotides. And so I know it says side here, but it really is a tide. And you can have nucleotide monophosphate, triphosphate, diphosphate. And then this is just to show you the differences. So for our purines, we of course have adenine and guanine, and then our pyrimidines, we of course have our other bases, cytosine, thymine in our DNA, and then uracil will replace thymine in our RNA. Now, pyrimidine bases are going to be synthesized as free bases, and then they're going to be converted to a nucleotide. And we'll talk about the synthesis of pyrimidines in the next lecture. Okay, so I just went over that. All right, so now we're going to have the formation of purines. So keep in mind, purines, as well as pyrimidines, are going to have many functions. They're going to be precursors of DNA, RNA, as I said before. They also are going to have structural input into coenzymes, such as NAD, FAD, and coenzyme A. As you know, they're going to be critical for making things like ATP. So they give energy. They are going to be structures involved in actually forming the bases, as well as giving energy to other processes. They are also going to be second messengers, often in intracellular signaling, and they are going to be allosteric regulators. So we've talked about some of them being allosteric regulators of different enzymes. AMP, for example, is a regulator to shut off fatty acid synthesis, for example. Okay, so purine bases are going to be produced de novo, and this is the sort of just a sort of general pathway for producing these. Most of the synthesis is going to occur in the liver, and there are going to be the nitrogen bases and nucleosides that will be then transported by your red blood cells throughout the body. This de novo pathway is going to require 6 ATP per purine And so this is why it's important to have a salvage pathway um, in order for many cell types to convert the free bases and nucleosides back into nucleotides. And so you can see I have here the purine, the two rings that will be formed. And so you have your glutamine, you have your ribose 5-phosphate that is going to first get activated with ATP. And then your ribo 5-phosphate is going to become 5-phosphorybosyl 1-pyrophosphate. Oops. Okay, in this first step of your ribose 5-phosphate plus ATP is going to be activating the ribose 5-phosphate. And this requires ATP, of course, and the mutations in the gene PRPS1 are associated with two syndromes. It's called ART syndrome and Charcot-Marie-Tooth disease, which are kind of have a wide range of disease outcomes. The ART syndrome is going to be associated with profound sensorineural hearing loss, ataxia, and development delay. So, oops. I did that backwards, didn't I? Okay, so here again, we're going to go through this process. We'll go through the steps, but we're going to generate that first nucleotide, inosine monophosphate. And then via two pathways, we are going to eventually get to guanosine or adenosine. And of course, we can also make RNA with these as well as DNA. Okay, so again, the activation, the first step is going to be ribose 5-phosphate. This, of course, came from the pentose phosphate pathway, if you remember that pathway. And so the pentose phosphate pathway will give you your 5 carbon sugars, and then you're going to have synthesis or activation of your ribose with ATP, and this is going to form your 5-phosphorybosyl 1-pyrophosphate. The enzyme is going to be the phosphorylase 1-pyrophosphate synthetase, and this is feedback regulated. So it's feedback regulation. This is not the commitment step. For your purine synthesis. Okay, so once you have this this first PRPP, then you can start bringing in your other components to start developing your ring structures. And so the first committed step is going to be the inclusion of your nitrogen from glutamine. And so remember that this is a ribose, so keep in mind that this is your anomeric carbon, remember? So that's that carbon 1. So you're building off of that ribose carbon 1. And so you're going to add nitrogen donated from glutamine, and you're going to release your 2 phosphates. And this is going to generate your 5-phosphobeta ribosylamine. And this is the first commitment step. Now this nitrogen is going to be nitrogen 9 of the purine ring. So we're talking here, it's carbon 1 of your ribose here. We know it's ribose because of the OH. And then this is going to be your nitrogen 9. So it's this one right here that gets built first. So your nitrogen 9 is what actually binds to your ribose or your deoxyribose. And this too is going to have feedback regulation. It's going to be a highly regulated. And it's going to have feedback regulation. Okay, then the next step is you have your 9 here, we're still building, and then you're going to add an entire glycine molecule to this growing purine. The glycine molecule is going to provide your carbons shown here, 4, 5, and then nitrogen 7 of your purine ring. Excuse me. And so you have your nitrogen that came in from glutamine. Which gave you that nitrogen 9 off of your ribose. And then you have your glycine that is going to be transferred to this growing chain. So this is through a glycine synthetase. Okay, so then the next part is you're going to have your carbon 8, which is going to be donated by N10 formyl tetrahydrofolate. So we're over here. And so that's going to give you your carbon 8, and this is by a glycamide ribonucleotide transformylase. And so it just adds that that folate. And then you regenerate your tetrahydrofolate, your H4. All right, then after this, you're going to add another nitrogen from glutamine. And so now we are in this position. Oh, sorry, I messed up that one. That was the first one. We're adding nitrogen 3 now, so we're building off in this direction. And so this is going to be by a formyl glycamide ribonucleotide aminotransferase. This step also is going to require ATP, and you get air formed, which is down here. You get a 5-amino imidazole ribonucleotide formed, and the ring closes. Okay, so now we have this whole structure here formed. We started at 9, we went to all the way around, and we added this nitrogen, and then the rings closes. Okay, so just keep it in reference. This is position 9, and we have our 4, and we have our 5, we have our 7, and we have our 8. Okay. And so we are going to add now a carbon 6, and we are also going to add the carbon 2. So we have in this place, we have our air synthetase, and then we have our CAR mutase. And so the route that takes place in the liver is shown here. So this is the liver route. It has here a side step in this particular book of an air carboxylase. We don't use that route. That's going to be in other organisms, bacteria and things like that can use the air carboxylase. So that's not for our route. Our route is going to be using the N5 carboxyamidodiazole ribonucleotide synthase, and then the N5 carboxyamidodiazole ribonucleotide mutase. We're adding a carbon from, in this case, bicarbonate, and so that's going to be our carbon 6 that's going to form, so that's this one right here. And then we're also adding a carbon from our N10 formyl tetrahydrofolate, and that's going to come in next. So for the case of adding this carbon dioxide, you can see that we have 1 ATP used here, and that's going to help facilitate that carbon being added. After that carbon's added, the next step is going to be aspartate. So aspartate is going to come in and donate a nitrogen to this nitrogen 1 position. So color that pink. And so again, this is going to require ATP, and then you're going to get that nitrogen that's going to be added to that carbon that came from that bicarbonate. And that is going to be by another synthetase, which is named for the product, the N-succinyl 5-imidazole for carbamide ribonucleotide. We still have aspartate attached until we have our carbon by the second formyl tetrahydrofolate come in. So this succinyl 5-imidazole for carbamide ribonucleotide through a lyase will release this chunk here, which is going to become fumarate. So we're generating a fumarate again here. And then you're going to add by a transformylase your last carbon, your formyl from your N10 formyl tetrahydrofolate, regenerating tetrahydrofolate, and getting that last carbon on your position 2. You then have IMP synthase, and IMP synthase is going to basically close the loop and release water molecules. And so this took a total of 6 ATP required to synthesize this first nucleotide, which is, of course, IMP. IMP synthase is going to be regulated by feedback regulation as well. Okay, so what is IMP? IMP is going to be the first nucleotide. It is going to have the base hypoxanthine, and this is going to be binding to the ribose. We already talked about this. Nitrogen 9 and our anomeric carbon are bound together. And so this is our base that we produce. Now IMP can become AMP, and what's important to understand between AMP and GMP is that you're going to have different energy sources. So to make AMP, you're going to use GTP. And this is going to take 2 steps. In the first step, you're going to add another aspartate molecule to IMP, and then this is going to form the adenyl succinate. And this is very similar to what we saw in the urea cycle from the arginosuccinate synthase in the urea cycle. And then and of course it requires GTP, which is shown here. And then after this, you are going to release fumarate, and then this is going to form the AMP. Okay, and then synthesis of GMP requires energy from ATP, and so ATP actually comes in the second step of GMP synthesis. So you have your hypoxanthine base that's going to be oxidized, and this is by IMP dehydrogenase to produce xanthine. And then glutamine will then donate a nitrogen to form GMP. So here's glutamine coming in to give that nitrogen to form the GMP. So here we have our xanthine, XMP, xanthine monophosphate, and then the donation of your nitrogen is going to form glutamine. And of course this requires ATP. Alright, so all these regulated steps So that first step of activating your ribose 5-phosphate is going to be regulated by your ADP, adenosine diphosphate. And so this comes from that AMP that was formed from IMP. And then you have other downstream steps that are regulated. You have the next step, the aminotransferase, which is bringing in that glutamine. And this is regulated by all of them, AMP, GMP, IMP. So if there is an increase in any of those substrates, that will prevent more purines from being made. And then you have your 9 steps through your ribosylamine to IMP, and your IMP dehydrogenase or your adenosuccinate synthase to make AMP or GMP respectively is going to be feedback regulated. So GMP will obviously impact IMP dehydrogenase, and AMP will impact adenosuccinate synthetase. Okay, so all of this is going to be feedback regulations. So the 4 key enzymes that are regulated are going to be the ribose, phosphate pyrophosphate kinase, also the glutamine aminotransferase, and then we have our adenosuccinate synthase and our IMP dehydrogenase. And so basically, if you have too many GMPs, that's going to push things towards AMP. Too many AMPs are going to push things towards GMP. Too many of either of those or both of those are going to push things back to ribose 5-phosphate. Now the next step is going to be degrading the purine bases. So this is going to mostly occur again in the liver. And you have salvage enzymes that are going to be used for most of these reactions. So you have AMP that is going to be deaminated to produce IMP, and that's through AMP deaminase. And then IMP and GMP are going to be dephosphorylated, and the ribose is going to be cleaved from the base by the purine nucleoside pyrophosphate. Okay, so you have AMP, which is going to become IMP, and this is an AMP deaminase. And then you have your IMP and GMP get dephosphorylated, and then you're going to get cleavage of your ribose, release of these ribose here. And this is by a purine nucleoside phosphorylase. Can't fit that. Okay, and then you get your ribose released. And then you have your bases, you have guanine, hypoxanthine. And then the important one here is going to be xanthine oxidase. Xanthine oxidase is going to be a target for alpurinol, which is, of course, going to be one of the treatments for gout. And then you have xanthine that gets formed from either guanine or hypoxanthine being further degraded. Xanthine oxidase again twice, so this is the same. So this is maybe 1, and then here's the second step for xanthine oxidase. Will generate uric acid, and uric acid will be released and go to the urine. Now, xanthine oxidase to uric acid excretion in the urine, of course, if you have too much purines that are being degraded, you'll get an increase of uric acid. All right, so we're next going to talk about our purine salvage. So the synthesis of these purine nucleotides is going to mostly occur in the liver. The brain can also make some of the purines, And then your immune cells can also make some purines de novo. So I have more space here, then let me do that here. So de novo, and this is all what we've been talking about, occurs in the liver primarily. Also in the brain and in your immune cells. Okay, and so within the liver, your nucleotides will be converted back to nucleosides or free bases, and then these are going to be transferred to other tissues via the red blood cells. So in the liver, you get your nucleotides, which will then become either nucleosides or can become free bases. And these are going to be picked up your red blood cells and transported. And why you use red blood cells is because red blood cells go everywhere. So cells that are even at the ends of capillaries into your different tissues will get access to this nucleoside or free base pool. Now what happens is the bases will be picked up. So you have your cell, be anywhere, muscle cell, and it'll pick up your bases from your red blood cells. And then most of these cells are going to be able to salvage these bases to generate nucleotides. And then you can go to RNA or DNA. Okay, so this is very important because this salvage pathway is going to allow the bases, nucleosides, and nucleotides to be easily interconverted and also enable the liver, that is the major producer, to release these into the circulation so that cells can pick them up and salvage these bases. Now there are two key enzymes we're going to talk about. There's actually three. We'll bring up a third one. But we have two key that are adenosine phosphorylase, APRT, and the second is hypoxanthine guanine phosphorylase transferase. And so, of course, when we're talking about this, we're talking about guanine. When I'm talking about this one, we're talking about adenosine. So these are the two main purine bases. So both of them have salvaging enzymes. Now here we have an image from your book, which looks very busy, but it's pretty straightforward once you wrap your head about what's going on. So in order to start, you first start at the top. Here you have your free bases. You have adenine, hypoxanthine, guanine. Those are your free bases. And then in the middle here, you have your nucleotides. You have your AMP, IMP, GMP. And then over at the far end, you have your nucleosides. You have your adenosine, inosine, and guanosine. Put it up a little bit. There. Okay. And so basically what you're trying to do when you're salvaging is you're trying to go, in the case of a cell that picked up a free base, you want to make your nucleotides or you want to make nucleosides. And so a few enzymes are going to help you do this. So for adenine, you have the adenosine phosphorylase transferase. That's okay. Shown here. And this can take the free base adenine and add a phosphate to that to convert it to AMP. And then once it's in AMP, you can go in many directions. You can go to, you can go to adenosine, and then adenosine through another important enzyme, adenosine deaminase. Oops, wrong, sorry. Adenosine deaminase can reconvert AMP through adenosine. It reconverts inosine through adenosine. Now, why this is important is because once you get into inosine, inosine is sort of the gateway between the two AMP and GMPs. And so inosine is able to reconvert IMP, and then that can become GMP or AMP. So depending on what the cell needs, once you get into inosine, you can go into both directions, as we saw when we were forming either AMP or GMP. So adenosine deaminase is very important for that because if you get a backlog of adenosine, it enables to free that through this inosine pathway. Now, hypoxanthine and guanine are going to use that different enzyme that's important in salvaging. They're going to use the hypoxanthine guanine phosphorylase transferase. They both use the same enzyme. So guanine can use this HGPRT to make GMP. Your inosine through hypoxanthine can use this same enzyme to make IMP. And then you have different enzymes that will interconvert. So for adenosine, an adenosine kinase can regenerate AMP. For AMP, and let me make these a different color. For AMP, you have AMP deaminase, and that can lead to IMP. And then once you have IMP, you can go through that whole process that we talked about before using ATP to make GMP. You can also convert it back to a nucleoside inosine, and then you can have a purine nucleoside phosphorylase that regenerates hypoxanthine. For GMP, you have a 5' nucleotidase, and that can also use that same purine nucleoside phosphorylase that was used by IMP and regenerate guanine. So all of these enzymes are there just to move things around to make sure that you're not wasting your purine rings, that you're salvaging those by making all these intermediates and all these final products or whatever the cell actually needs. Okay, so again, you have your phosphorylase transferase, which is catalyzing the addition of the ribose 5-phosphate group from that PRPP, the activated ribose, to a free base. And this generates a nucleotide and a pyrophosphate. So you get your nucleotide from this as well as a pyrophosphate. As we saw before, adenosine and AMP can be deanimated by adenosine deaminase and AMP deaminase, and these are going to form inosine and IMP as we showed before. Now you have purine nucleoside phosphorylase, which is going to catalyze a reaction in which the glycosidic bond that is attaching to the base of the sugar in guanosine and inosine. And so that is right here. And so this is going to generate ribose 1-phosphate as well as release a free base. And guanosine and inosine are converted to guanine and hypoxanthine in this manner. So this is using the purine nucleoside phosphorylase. So what's the clinical significance of this? And again, this takes a lot of energy to make your purines. A lot of diseases are associated with one even making that first activated ribose, as we talked about, Art's disease, for example. Also, there are other diseases involved in the salvaging of purine bases. And these different diseases can lead to very mild or fatal disorders. So I'll just go through a few of these. So Nyam syndrome is going to be a lack of that hypoxanthine guanine phosphorylase transferase activity. So remember that this was that was the salvage enzyme for both hypoxanthine, which came from inosine to make IMP, and then also from guanine to make GMP. So this is very important. This leads to a lack of salvage pathways. What that means is that you have to make all your purines by de novo synthesis. And so early on in development, children typically by the age of 2 have intellectual disabilities, and they also show self-destructive tendencies. So they mutilate themselves. They bite their fingers and toes and their lips. It's a very debilitating disease. Another is that ADA deficiency. So if you remember, that's the adenosine deaminase deficiency. This is going to lead to ADA, severe combined immunodeficiency syndrome. This is autosomal recessive, so it's very rare. ADA is going to eliminate deoxyadenosine, which gets generated when DNA is broken. And then the ADA will convert the deoxyadenosine to deoxyinosine, which is harmless. And so ADA deficiency does not allow you to do this conversion, and so you block that, and you are going to basically lead to an increase of deoxyadenosine, and that is toxic to cells. This is going to be an immunodeficiency syndrome. It's primarily going to affect cells that are rapidly dividing, such as your immune system. And so what this basically does is your immune system, as it's developing, the cells are dying because they're building up this deoxyadenosine. Why it's called severe is because you lose all your immune cells. You lose your B cells, you lose your T cells, you can't make antibodies, etc. And so it's a SCID, severe combined immunodeficiency syndrome. There are other forms of SCID. So this one is specifically adenosine deaminase deficiency associated SCID. So SCID, as I said before, loss of T and B cell functions. There are going to be different causes. We're going to talk about more of this in immunology. You can have loss of function of different receptors or different communicators of the immune system. You don't have to worry about that for this particular test. Again, we'll do this in immunology. But what you do have to know is that 15% are caused by defects in that enzyme we just talked about. You're going to destroy your B and T cells. B cells make antibodies. T cells are T cell responses. So you have a loss of your immune system, basically. So one thing that helps people understand what this means is this was sort of that bubble boy syndrome. People have to live in a sterile environment because they can't be exposed to any potential microbe, because even the most benign microbe can kill a person with SCID. You are going to accumulate your deoxyadenosine, and this is going to trap everything in that deoxyadenosine, so you don't get that salvaging, you don't get the other nucleotides being produced. Because of this lack of nucleotides, you inhibit DNA synthesis, and your immune cells basically cannot survive. There's also von Uric disease, and this is glycogen storage disease. The deficiency is going to be associated with glucose-6 phosphatase activity. And remember that you have glucose-6 phosphate pushes things through the pentose phosphate pathway, and the pentose phosphate pathway is going to give you your 5 carbon sugars. And so your increased levels of your 5 carbon sugars will increase the activated ribose. And this increase leads to an increase of purine synthesis. And then this purine synthesis leads to an increase in degradation. Which then leads to an increase of uric acid. Now, the excess of uric acid doesn't necessarily have to be caused by deficiency. It can be caused by diet, but an excess of uric acid is going to ultimately deposit crystals in your joints and kidneys. And what happens when anything gets deposited somewhere, your immune system thinks that it's foreign and it attacks it. So this is going to cause inflammation by your immune system. So treatment of this, if it's diet related, is to reduce high foods nucleotides. And also allopurinol is going to inhibit that xanthine oxidase, and that's important in converting these purines to uric acid. So allopurinol is a substrate of xanthine oxidase, And so allopurinol is a competitor for your purine's access to xanthine oxidase. Also, it is a inhibitor because it binds to the actual enzyme, so it'll bind to xanthine oxidase and not allow xanthine oxidase to form uric acid. So allopurinol will compete with your purine degradations and form oxypurinol, and then that can easily be excreted. Now, muscles, of course, are going to, when you're having some sort of energy, or you're going to increase the demand for energy, and a main source of energy in muscles is to break down your branched-chain amino acids. The nitrogen that gets transferred to aspartate in this will enter the purine nucleotide cycle, because remember, aspartate was donating nitrogens in this cycle in a few spots. So in the muscles, you have AMP deaminase, shown here. And this is going to be called in the muscles myoadenylate deaminase. Let's write it here. So in branched chain amino acid catabolism, down here we have branched chain amino acid catabolism. It's going to release nitrogens that get transferred to aspartate. And that's through just a general transamination reaction that we've talked about, glutamate donating the nitrogen to aspartate. And then aspartate is going to get deaminated to fumarate. And then the nitrogen is going to be acquired by glutamate to form glutamine. Fumarate is used, of course, in the TCA cycle to replenish the cycle to maintain high levels of energy as needed. Now, aspartate is going to do this through an intermediate, the adenyl-succinate intermediate, and then adenyl-succinate will form fumarate, and you're also feeding this into the purine nucleotide cycle. So you're going to donate that nitrogen, form fumarate, as we saw in our previous slides, and that's the role of aspartate in the purine nucleotide cycle. Okay, so I'm going to stop here, and our next lecture will be on the synthesis of pyrimidine bases.